Welcome friends. The topic for discussion in this video is insulin synthesis and insulin secretion. I am Dr. Omkar Singh. Insulin is synthesized within the beta cells of islet of Langerhans. Insulin gene is located on chromosome 11, very close to the gene for insulin-like growth factor 2. The insulin messenger RNA is translated as a single chain precursor known as pre-pro-insulin. It is a 110 amino acid polypeptide. It contains an amino terminal signal sequence followed by the B chain, a connecting peptide, and a carboxy terminus A chain. The signal peptide interacts with signal recognition particles. Signal recognition particles are cytosolic ribonucleoproteins. Function of these signal recognition particles is to facilitate the translocation of pre-pro insulin through the membrane of endoplasmic reticulum into the lumen. During insertion into the endoplasmic reticulum, the signal peptide is cleaved by a signal peptidase to yield the proinsulin. Proinsulin then undergoes folding and the formation of 3 disulfide bonds within the lumen of endoplasmic reticulum. The folded proinsulin then enters into the Golgi apparatus, where it is packaged into secretory vesicles. Within the vesicles, it is cleaved by specific endopeptidases to yield this insulin molecule and the connecting peptide in equimolar concentrations. The insulin molecule, connecting peptide, and the amylin molecules, they are stored in these vesicles. This is the structure of a mature insulin molecule. It contains 51 amino acids and is a heterodimer consisting of an A chain with 21 amino acids and a B chain with 30 amino acids. The two chains are linked to each other by 2 disulfide bonds. In addition, there is an additional disulfide bond present within the A chain. Insulin has 3 conformational states: monomeric state, dimeric state, and the hexameric state. The state of insulin is dictated by the concentration of insulin as well as the pH of the surrounding fluids. As the concentration of insulin rises, insulin monomers tend to form dimers, and in the presence of zinc and favorable pH, the monomers will assemble into hexamers. The hexamers secreted from the beta cells, they diffuse into the bloodstream down their concentration gradient. A combination of electrostatic repulsion and decreased concentration of insulin favors the dissociation of insulin hexamers into insulin monomers. Insulin monomers are the active form of insulin, while insulin hexamers are the storage form of insulin. Insulin is the only hormone that lowers the plasma glucose concentration, and glucose homeostasis is maintained as a result of regulated insulin secretion. Pancreatic beta cells monitor the extracellular glucose concentration and secrete insulin as required at a given time. The beta cells are equipped with multiple sensors that measure the circulating glucose. These include GLUT2 channels. In addition, the enzyme hexokinase also functions as an insulin sensor, and The ATP ADP ratio, which regulate these ATP gated potassium channels, also can monitor the blood glucose concentration present in the extracellular fluid. Coming to the glucose induced insulin secretion, glucose is transported into the beta cells through these GLUT2 transporter channels, and this occurs through facilitated diffusion. GLUT2 is a glucose sensor in the beta cells. Mobilization of the GLUT2 to the plasma membrane is insulin independent, and the GLUT2 transporter protein shows a low substrate affinity, ensuring high glucose influx into the beta cells. That means at low blood glucose levels, very little glucose will enter into the beta cells. At high blood glucose concentration, large quantities of glucose will enter into the beta cells. Two important properties enable glucose kinase to function as a glucose sensor in the beta cells. Once the glucose has entered into the beta cell through the GLUT2 channels, the glucose gets phosphorylated by the enzyme glucokinase. This glucokinase enzyme has a lower affinity for glucokinase compared to other hexokinases. Another thing is that glucokinase is not inhibited by its product, which is glucose 6-phosphate. That means the continued activity of the enzyme in spite of high glycolysis load. Glucose kinase is thus a rate limiting step in beta cell glucose metabolism, and it is considered to be an important glucose sensor. Glucose 6-phosphate enters the glycolytic pathway and then into the Krebs cycle, where multiple ATP molecules are generated by the oxidation of acetyl coenzyme A, leading to a rise in the ATP-ADP ratio within the beta cells. On the cell membrane of these beta cells are present ATP-sensitive potassium channels. An increased intracellular ATP-ADP ratio will close these potassium channels at low glucose concentration levels. These ATP-sensitive potassium channels are open, and positively charged potassium ions can move out of the cell, maintaining the negativity inside the cell. However, increased formation of ATP when blood glucose concentration is high will result in the closure of these ATP-sensitive potassium channels. Potassium ions which are positively charged will not be able to move out of the cell, resulting in increased positivity inside the cell. This condition is called depolarization of the cell, and this will cause the inside of the cell to become less negative with respect to the outside, leading to the depolarization of the beta cell membrane. Another channels which are depolarization sensitive calcium channels are present on the cell membrane of the beta cells, and when there is high concentration of ATP within the beta cells, they result in the depolarization of the cell membrane and lead to the opening of depolarization sensitive calcium channels. The resulting increased intracellular calcium concentration causes the release into blood previously synthesized insulin, which has been stored in the intracellular secretory vesicles through the process of exocytosis. The glucose stimulated insulin secretion can be modified by the incretin effect. This incretin effect describes the observation that orally but not intravenously administered glucose enhances insulin secretion. The resulting potentiation of the insulin secretion may account for up to 50% of the total insulin release. Incretins are gut-derived hormones which potentiate insulin secretion after ingestion of meal, and this occurs in a glucose dependent manner. The two best studied incretins are glucose dependent insulinotropic polypeptide abbreviated as GIP and glucagon like polypeptide 1 abbreviated as GLP-1. Incretins exert their insulinotropic actions through G-protein coupled receptors which are highly expressed on the beta cells. Incretins have no effect on ATP gated potassium channels. The incretins bind to the G-protein coupled receptors on the beta cell membrane and increase the intracellular cyclic AMP concentration. The cyclic AMP enhances glucose stimulated insulin secretion through activation of phosphokinase A, which phosphorylates proteins which are involved in insulin exocytosis. So, the glucose stimulated insulin secretion can also be mediated by many other factors, including non-glucose nutrients, hormones, and neural inputs. One of the important non-glucose nutrients are the free fatty acids, and they participate in the regulation of glucose stimulated insulin secretion. Insulin secretion is suppressed with long-term exposure of beta cells to excessively high concentration of fatty acids, and this condition is called lipotoxicity. That means when the beta cells are exposed to high concentration of free fatty acids over prolonged period, the glucose stimulated insulin secretion may be depressed, and this condition is known as lipotoxicity, and it will result in insulin resistance. On the other hand, short-term exposure to the physiological concentrations of free fatty acids causes time-dependent potentiation of the insulin secretion. The insulinotropic action of the free fatty acids, when these are present in physiological doses for short duration of time, these are mediated by membrane receptors for free fatty acids, and these membrane receptors for free fatty acids are GPR40 receptor molecules. The activated receptor elevates the calcium ion concentration within the beta cells through activation of the phospholipase C enzyme and the protein kinase D1. Free fatty acid augmentation of glucose stimulated insulin secretion is independent of ATP sensitive potassium channels. The parasympathetic stimulation of pancreatic islets operates via the activation of phospholipase C. Binding of the acetylcholine to a G protein coupled receptor within the pancreatic islets activates the enzyme phospholipase C, resulting in increased insulin secretion into the blood. The phospholipase C cleaves the membrane phospholipid inositol biphosphate into PIP3 and diacylglycerol. The IP3 then binds to a receptor protein in the membrane of endoplasmic reticulum. The binding of the IP3 to the receptors in the endoplasmic reticulum allows the release of calcium, and this raises the intracellular calcium ion concentration, and this effect is independent of the effect of high blood glucose on calcium ion concentration within the beta cell. Another nutrient, which are the amino acids, they can promote insulin secretion. The individual amino acids at physiological concentrations are poor insulin secretagogues. When present alone and at physiological doses, individual amino acids do not increase the secretion of insulin. However, in certain combinations of amino acids at physiological concentrations, they can augment the glucose stimulated insulin secretion. For example, glutamine when present alone does not enhance glucose stimulated insulin secretion. But a combination of glutamine with the another amino acid leucine can enhance glucose stimulated insulin secretion from the beta cells. This amino acid leucine activates the enzyme glutamate dehydrogenase. The activated glutamate dehydrogenase enzyme converts glutamate into alpha ketoglutarate. This molecule enters into the TCA cycle, which results in increased ATP production, thereby enhancing the insulin secretion. Thanks. Have a good day.